A minute for everyone to get in. Okay, I'm going to start the introduction of this seminar. So welcome everyone for this third GREG seminar. I'm Anne Charmentier and I'm president-elect for the SSE for 2023. We welcome all SSE members and beyond to discover the amazing research of PhD students that were recipients of graduate research excellence grants by the SSE. This series of seminars is organized by the Graduate Student Advisory Committee of SSE GSAC, and I want to thank them for what is a huge amount of work around the GREG Awards and seminars. If you're interested in the SSE GREG Awards, you can follow the link that I will post on the chat right after the introduction, and that explains what these awards are about. So today, it's my very great pleasure to introduce our GREG seminar speaker, Sage Madden. Sage is a third-year ecology PhD student in the lab of Dr. Gail Patricellis at the University of California, Davis. Her research interests revolve around the behavioral and physiological responses of animals to human-induced environmental changes, and especially urbanization, and to study whether these responses are adaptive. Sage is also involved in efforts to engage the public in the scientific process as a community and citizen science and conservation fellow at the UCD Center for Community and Citizen Science, and to recruit and retain diverse voices in STEM throughout K-12 outreach and mentorship of undergraduate students. Today, Sage will tell us about Do Urban Songbirds Beat the Heat? That's the title, with the subtitle Effects of Urbanization and Heat on the Behavior, Physiology, and Fitness of Black Phoebes. Now, Sage's seminar will last 45 minutes, 40 to 45 minutes, and then we will have 15 minutes of a question session. We expect everyone to follow the code of conduct for both during the seminar and the question session, the link of which I will also place on the chat right after this introduction. So Sage, I'm very eager to hear about your research now. The floor is yours.
Awesome, thanks. Sorry, just finding the unmute button with the screen shared. Are you seeing presenter view or are you seeing
It's perfect, and we're hearing you well as well. It's great.
Awesome. Okay, I'll go ahead and get started. Thank you for that introduction. Today I'll be talking to you about my dissertation research on the interactive effects of urbanization and temperature on a songbird called the black phoebe, with a special focus on the evolutionary aspects. All right. So I'll start by just giving you a quick overview of where this talk is going to go. I'm going to start with some conceptual background on the effects of urbanization and temperature, as well as the hypotheses I'll be testing. Then I'll give you an overview of my research specifically and my study system, and I'll talk you through 2 of my 3 dissertation chapters, and these are the 2 that are the most relevant to the evolutionary dimensions of the work that SSE has helped fund. And finally, I'll end with some next steps and significance. So starting with the conceptual background, we know that land use change, including urbanization and climate change, are both key drivers of biodiversity change. But to date, their effects on organisms have typically been studied in isolation. So we're either looking at the impact of land use change or the impact of climate change on biodiversity separately, which leaves some pretty important unanswered questions, including whether urban organisms respond differently than non-urban organisms to climate change-driven temperature extremes. So when we look at these two effects in isolation, we don't know whether when they combine, as they often do in the real world, we'll see a combined effect that's equal just to the sum of the two separate effects, which we would call an additive effect, or whether we might see that when we combine the two, they actually have stronger effects on biodiversity or other processes than we'd expect simply from summing them, and we call that a synergistic interaction. Or finally, whether they might actually have weaker impacts in combination than we would expect just by summing their separate effects, and that would be an antagonistic interaction. Just to start with some pretty cool examples suggesting that there may, in some cases, be interactive effects of urbanization and heat on organisms. On the left here, I'm presenting a study on great tit reproduction. And this plot shows measures of nestling morphology and then nestling mortality on the y-axis, and then number of hot days on the x-axis. And what we're seeing here is that in these blue forest sites, growth declines as the number of hot days increases, whereas in the red urban sites, it seems like growth doesn't change much or maybe even increases. And similarly, it seems like there might be different effects of heat on survival in these urban and non-urban nestlings. Likewise, another really cool example is in crested anoles. And here you're seeing on the y-axis the critical thermal maximum, which is a measure of an organism's thermal tolerance. And on the x-axis, you're seeing and within each region, the authors collected anoles from a forest site and an urban site. Urban is in the open circle here. And what this plot is showing is that crested anoles that were collected at urban sites had higher heat tolerance than those collected at non-urban sites. So another cool example of potential differences between urban and non-urban animals in their responses to heat. But why might we see these differences? I'm going to explore sort of three categories of combined effects. The first is thermal effects. Second, resource and cost differences between urban and non-urban environments. And the third is evolutionary and developmental effects of the urban environment. And I'll go through these in more detail now. So starting with thermal impacts, we know that temperatures in cities are often higher than those in nearby areas. That's what you're seeing on this plot here. On the y-axis, we have temperatures after sunset, and then on the x-axis, we have a variety of different locations along an urbanization gradient. And what we see is that temperatures peak at more urbanized locations. And this is strongest at night, but often also happens during the day. And we call this the urban heat island effect. Likewise, climate change is generally expected to lead to higher mean ambient temperatures, greater variability in temperature, and more frequent extreme weather events, including heat waves. And interestingly, when we combine climate change and the urban heat island effect, they sometimes have interactive effects on the thermal environment. So what this plot is showing is the difference in the urban heat island during a heat wave versus a normal day. And a filled circle is indicating that the urban heat island is stronger during a heat wave than it is on a normal day, whereas the open circle indicates the opposite. And so what we see is that in current climate in many cities, heat waves and urban heat islands interact synergistically such that temperatures are hotter than we'd expect simply from their combined effects. And so these synergistic interactions might lead to urban organisms being more likely to reach some kind of thermal limit because they're just experiencing way hotter temperatures in cities. And that might lead to stronger effects of temperature on fitness in cities. Pipoli et al. Called this the superimposed urban heat wave and heat waves hypothesis. Moving on to the next category of impact, we know that urbanization alters resource availability and costs of living for a variety of organisms. So for some organisms, including insectivores and breeding birds, food availability and quality are likely to be lower in cities, as insect abundance and diversity often declines with urbanization. But for other animals, food availability might actually be higher because they have access to human trash or food subsidies like bird seed. Likewise, water availability may be higher in cities for many organisms because cities often experience higher precipitation than nearby areas, and they also have artificial watering in many cases. but water quality is often lower. Water sources in cities are often more polluted and contain a lot of trash. Finally, cities might differ in the availability or quality of thermally buffered microsites, basically like shaded sites where animals can seek refuge from high temperatures. But the way that they differ likely depends on what type of natural habitat we're comparing them to. So you could imagine that a forest might have more microsites and better quality microsites than a city, but a city might have more than, say, a grassland. Moving on to costs, we know that the concentrations of many chemical pollutants, as well as noise and light pollution, are often higher in cities. And for some species, there may be a higher abundance of predators and parasites in cities. And for example, for songbirds, there's often higher densities of nest predators and predators of adults like domestic cats in cities. And these may combine to lead to both energetic and opportunity costs in cities. So we know that things like exposure to chemical pollutants and noise levels cause physiological stress and require increased energetic expenditure. But likewise, they might disrupt foraging and cause an animal to allocate more time and energy to things like thermoregulation and vigilance at the expense of foraging or caring for their young, which I'd call an opportunity cost. So I'm going to talk briefly about a framework that uses energy and opportunity, or energy and costs, to understand organisms' resistance to perturbations like extreme climatic events. And this is John Wingfield's framework. So what you're seeing on the y-axis here are different variables that relate to energy. On the x-axis, this is time across the season. And here, ECR is the cumulative resources an animal has access to. This changes throughout the season. EI is an animal's costs of going about its daily activities. And EE is the costs of just living and existing. And so Wingfield proposes that an animal's ability to cope with stressors or perturbations like heat waves depends on how much energy it has in relation to the cost it experiences. So if it has more energy compared to costs, it has higher perturbation resistance potential. And if it has less energy compared to costs, it has lower perturbation resistance potential. I've just finished telling you that for many organisms, likely including songbirds, there is decreased resource availability and increased costs in urban environments. And so here we're decreasing cumulative resources, we're increasing costs, and the combination of those would decrease the organism's perturbation resistance potential, which might lead to extreme temperatures being more likely to put the animal in a state we called allostatic overload, which is basically when costs exceed resources. And when an organism enters this state, it often goes into what Wingfield calls an emergency life history stage, where it's basically abandoning effort into reproduction or whatever stage it's currently in, and just focusing on survival. And I'd call this the urban poor resources high cost hypothesis. Finally, considering the third lens, up to this point, I've been talking about just an organism at the current time point that's living in an urban environment and how it's experiencing that environment. But if we add in the component of time and consider that many organisms have been living in urban environments for decades or centuries, then we might actually expect that urban organisms would be more tolerant of extreme temperatures due to evolutionary or developmental changes in their traits that have occurred through their exposure to the urban heat island. And Pipoli et al. Call this the adaptive responses to urban heat island hypothesis. And so just to give some more detail and examples, we might, for example, expect that natural selection due to exposure to this urban heat island might lead to genetic changes that alter an organism's tolerance of temperature. And returning to that enol example I gave earlier, the authors actually found that there was a single nucleotide polymorphism that occurred at higher rates in city organisms and was associated with higher thermal plasticity and so likely greater heat tolerance in these urban anoles. So that's a cool case of genetic differences between urban and non-urban organisms that relate to their heat tolerance. If we also consider developmental effects, we might expect that exposure to extreme temperatures during development might prime an organism to respond to those temperatures as adults. So basically make it better able to respond to those temperatures. And that's what this plot on the right is showing. So we have thermoregulatory performance on the y-axis, and we have the adult's thermal environment on the x-axis. And the color here is the thermal environment that the organism developed in. So what we're seeing is that an organism that developed in a cold environment performs better in that cold environment as an adult, whereas an organism that developed in a hot environment performs better in a hot environment as adults. So an organism that developed in a hot urban environment might perform better in hot environments as an adult. On the other hand, as kind of an alternative to this adaptive responses to urban heat islands, we might actually expect that exposure to hot temperatures disrupts development and leads to reduced ability to cope with extreme temperatures as an adult. So that's what this second plot facet is showing. Basically, an organism here that developed in a normal environment has an advantage over organisms that developed in extreme environments, regardless of the adult thermal environment. So to give a quick recap of these three lenses, first, I might expect that because of the combination of the urban heat island and heat waves or climate change-induced temperature change, urban organisms are more likely to reach thermal limits. So we might see stronger negative effects of temperature on fitness and traits in cities. From the second lens, urbanization leads to changes in resource availability and costs. And for some organisms that experience decreased resource availability and increased costs, urban organisms may have lower perturbation resistance potential, which may lead to stronger negative effects of temperature on their traits and fitness once again. And finally, from the evolutionary developmental lens, over time in urban environments, organisms may come to have differences in their traits, like, for example, physiological or behavioral responses to temperature, which might lead to weaker negative effects of temperature on their fitness in cities. From here on out, I'm going to focus on how two of my dissertation chapters provide insight into this third lens. But I want to note really quickly before moving on that my chapters do attempt to address all three of these, and you might expect that they map onto each a chapter. That's not the case. Insight from each chapter is used to address each of these. And that's because my chapters are organized around the type of data or the response variable. So just to prepare you for that. Okay, so now I'll move on to giving you an overview of my research and study system. My three chapters are first to examine how do urbanization and temperature interact to affect songbird reproductive success using black phoebe, which is this adorable little bird here. Second, again in black phoebes, to look at what behavioral and physiological mechanisms underlie the impacts of urbanization and temperature on reproductive success. And third, the final chapter, which I'm not going to talk about in detail here, is to use a continental data set of nesting success to look at what features of cities or species modify the nature of these interactions. And I've chosen to focus on songbirds here because eggs and nestlings of songbirds are particularly vulnerable to temperature fluctuations. Nestlings, when they first hatch, are ectothermic, which means they can't maintain a constant body temperature in the face of fluctuating environmental temperatures, which makes them very vulnerable to extreme heat. They do develop endothermy as they grow, and so as older chicks, like these pictured here, they have to deal with the costs of thermoregulation. Another really interesting feature of songbirds is that parents provide all of the energy resources the nestlings have access to, and so parent behavior determines their energy reserves for thermoregulation. And so although nestlings can't behaviorally escape temperature, really, their parents' behavior might determine how they respond to temperature. And finally, these birds provide a tractable system because they're diurnal. They're pretty easy to observe, and we can monitor nests to get a good idea of reproductive success. I specifically focus on a songbird called the black phoebe, which is this bird on the left here. These birds are a non-migratory insectivorous flycatcher, and they are common in urban and riparian areas in the Central Valley of California where I work, which is one of the main reasons I've chosen them as my study system. You can see on the bottom here a map of eBird, which is a community science platform to report bird sightings. And these are eBird sightings of black phoebes in the area that I work. So University of California, Davis is right here. And this here is the capital of California, Sacramento. And we have some riparian corridors running along here and then up here. And this is a wetland area. So you see black phoebes are pretty abundant in urban and non-urban areas. They're also a mud cup nester. So they build little open cup nests out of mud, which means that they might have less thermal buffering than, say, a cavity nester. And they're also less well studied than cavity nesting birds in studies of both urbanization and heat. And finally, it's relevant to note that these birds exhibit biparental care, which means that both mom and dad provide care for the chicks. All right, so I'll talk now through the questions and methods of my first chapter, as well as a sneak peek into some preliminary results. So returning to this example I gave you earlier in the presentation that found high temperatures had stronger effects on reproduction of great tits in forests than cities. This is a really cool pattern that kind of inspired my study. But just from the results of this study alone, we don't know what specific aspects of the urban environment are driving these differences in growth and survival. And we also don't know whether there are physiological or behavioral mechanisms driving these differences. And so in chapter 1, I first asked, do temperature and urbanization interact to affect reproductive success in phoebes? Basically, am I going to find this same pattern in my study system? And second, what features of the urban environment specifically are contributing to these interactive effects? So I'm doing 3 field seasons, one of which has already happened, then I'll do 2 more. And my field seasons are pretty long. They last from February to August. So here I'm showing you some monitored nests from my first season. On the right, you can see that my study sites are generally in the vicinity of Sacramento. So this is Sacramento here in this map. And I've got study nests in 4 cities, Sacramento, West Sacramento, Davis, and Woodland, and then in some wetland and riparian areas nearby. And so these are my nests that I found and monitored in my first season. I'm planning to add more nests in this most urbanized region of Sacramento in the next 2 seasons. And the core method of my first chapter is monitoring nests. So I'm showing you here a timeline of nesting phenology from the clutch initiation, which is the day the first egg is laid, to the hatch date, which is when the nestlings hatch, and at that point they're little gummy bears looking like this, to their fledge date, which is when they leave the nest. And here I've marked their day 14, which is basically the last day that I can safely handle them. If I come after this, I risk scaring them out of the nest. So we are visiting the nest and checking its contents using a mirror every 3 to 5 days from when the first egg is laid until when they're banded. And we're taking nestling measures of their mass and wing morphology at each of those time points during nestling development. On day 14, we take more detailed morphological and physiological measures that I'll talk about more in chapter 2. And from this, we get my fitness proxies, which are first nestling survival, which is pretty clearly linked to the reproductive success of the adult birds, as well as to the fitness of the nestlings. And nestling growth. Nestling growth is relevant because we know that the size at which a nestling leaves the nest is a strong predictor of how likely it is to survive and recruit into the population. So bigger nestlings are much more likely to survive their first year and to recruit. My first chapter also involves taking a variety of habitat measures. We're placing temperature sensors next to and inside each of my nest cups, and those are monitoring temperature from during the egg stage until the last time we handle the birds. I'm also taking measures of things like the predator and pollutant environment, including sensory pollutants and chemical pollutants. I'm not going to go into too much detail on that here, but happy to talk more if you're interested. And importantly, I am taking measures of the habitat composition surrounding the nest. And so here is an example of one of our nests at a park in one of the cities. And my measure of urbanization level here is the percent impervious surface surrounding the nest in some buffer. So I would look at how much of the buffer around this nest is made up of surfaces like roads and buildings to measure my urbanization level. And I want to note that I'm using this urbanization level measure in addition to a bunch of other measures of the urban environment. So in addition to looking at these sort of pan-urban effects using this percent impervious surface, I'm also teasing apart what other aspects of the urban environment are driving any of the effects I see. I want to, before moving on, briefly note also my statistical approach, which I think is relevant because I'm working on observational data in a pretty complicated system with a lot of correlated and interacting factors. And so I'm using generalized linear mixed models informed by causal graphs. So specifically, I'm focusing on Bayesian structural causal modeling to tease apart these relationships. And this is just like a super simplified example of one of the causal graphs where I'm looking at how urbanization level interacts with temperature to affect nestling growth and survival, while including other potential covariates or confounding variables, and also including random intercepts to account for non-independence among, like, for example, measures of nestlings from the same nest. So moving on to my predictions for chapter 1, I'm focusing on this evolutionary hypothesis. So if the adaptive responses to urban heat island hypothesis is true, then I would expect that at higher urbanization levels, I see higher nest temperatures. So basically here I'm saying I would expect to find an urban heat island. I would also expect that nest temperature has weaker impacts on reproductive success at high urbanization levels than it does at low urbanization levels. So at high urbanization levels, I would expect that higher temperatures lead to a less of a decrease in growth or survival than they do at low urbanization levels, which is the same pattern that I showed you earlier with great tits. I'm waiting to run the models for my analysis until I've collected my next few field seasons of data. But just to show you the variation that's present in my study sites here, I've got the daily maximum temperature at the nest across the nesting period summarized by different regions. So these are my different cities and nature preserves. And then on the right, I've got minimum temperature in the same regions. And just note the axes are on different scales here. And so what I'm seeing here is that there's a lot of variation in temperature both within my regions and across regions. The regions on the left are the ones with the highest impervious surface cover. The ones on the right have lowest. So generally, seems like temperatures are hotter at more urbanized sites. And strikingly, temperatures reach levels as high as 125 degrees Fahrenheit, or around 52, 53 Celsius. And no, the birds did not survive those temperatures. They also reach minimums as low as 39 Fahrenheit. And here you see there's not as clear of a pattern in minimums across my sites, but still a lot of variation. Here I'm showing you nest temperatures across impervious surface within 50 meters of the nest. And so it generally looks like the maximum temperature at the nest increases with percent impervious surface, and there's a good spread in both of these variables. But again, I'm waiting to run models until I have collected my remaining data. Here you see nest minimum temperature and the interquartile range of temperature, which is a measure of the variability. Finally, there is variation in nestling mass and the number of fledglings across my nests and sites. And so here I'm showing you nestling mass at banding for the 3 most common ages. We band them when the oldest nestling is 14 days old, but they don't all hatch at the same time, so they do vary in age when we band them. You can see that their mass at banding varies from anywhere around 12 grams to around 20 grams, so that's pretty striking variation. And likewise, a lot of our nests don't fledge any chicks. Some of them do, and within those nests, there's variation in the number that fledge. To show you a visual representation of the variation, here I've got pictures of two nestlings on their banding day. These birds are the same age and they were banded on the same day of the year, but you can see that the one on the left here is much smaller and less well developed than the one on the right. Moving on to chapter 2. I want to return again to this cool example in Great Tits, where high temperatures have stronger impacts on reproduction in forests than cities. And again, note that this is a super cool pattern, but we don't know what mechanisms are driving this. We don't know, for example, whether there are changes in the behavior and physiology of urban birds that might be driving this reduced impact of temperature. And so in chapter 2, I ask, does urbanization level affect the physiological and behavioral responses of my birds to temperature? How are these physiological and behavioral responses linked to their fitness, specifically reproductive success here? And finally, is variation in behavior across temperature and urbanization driven by phenotypic flexibility at the population level or by consistent individual differences in behavioral traits? And so here I'm trying to tease apart the sources of behavioral variation in my birds. I focus on nestling stress physiology and parental care behavior for this chapter because they are likely to mediate the impacts of temperature on reproduction. So if I consider how temperature impacts nestling growth and survival, one of the main pathways is likely that high temperatures affect energy balance and temperature balance for the nestlings. If temperatures are pretty high, nestlings might experience hyperthermia, and they're having to devote more energy to thermoregulation. They also might be experiencing cell damage or even mortality at really high temperatures, and all of this is expected to lead to elevated stress hormone levels. And to lead to reduced nestling growth and survival. Nestlings are diverting energy away from growth. And again, really high temperatures may just lead to mortality. A second pathway through which temperature might affect growth is that temperature affects the food availability for insectivorous birds, might affect both the abundance and diversity of insects, as well as the behavior of the parents. And so parents are also experiencing these hot temperatures, and they might be devoting more time and energy to thermoregulation. They might be perching in the shade instead of caring for their chicks. And so this lower food availability and greater parent time and energy allocation to thermoregulation might combine to reduce the amount of food the parents deliver to the chicks, which would again reduce their growth and survival. So these are kind of two hypothesized mediators of our effects of temperature on growth and survival, and so they're both very relevant to the fitness of these birds. But actually, their role as mediators is itself pretty understudied at this point, and so a lot of people are starting to examine that, including me. So to measure nestling stress physiology, I focus on measures taken during day 14 banding, because by this time I expect my nestlings to be old enough to have a developed HBA axis, so a developed stress response to acute stressors, and because they're old enough for me to safely collect a blood sample. So I'm taking what are called stress series blood samples. So I want to direct you to this plot real quick and point out that after exposure to some acute stressor, like me taking these birds out of their nest to measure them, we expect that stress hormone levels, called glucocorticoid hormones, will rise quickly and then fall. And so I'm taking two blood samples, one within 3 minutes of capture, which is represented by this little spot here, which measures the baseline level of these hormones in the nestlings, or as close to baseline as we can get. And then one about 30 minutes after capture, which represents the peak of this response to an acute stressor. So it lets me get at the responsiveness of this bird's HPA axis to this capture stressor. In addition to measuring glucocorticoid hormones, or CORT, I'm also measuring blood glucose. Glucose is mobilized as part of the stress response, and it's also affected by the bird's nutrition and insulin levels. So it's another interesting physiological measure. I'm estimating hematocrit, which is the percentage of the blood volume that's made up by red blood cells. So here you're seeing a tube of bird blood that's been spun in a centrifuge, and the red is the red blood cells, and the yellow here is the plasma. And so I'm looking at what percent of the volume of this blood is red blood cells. And that's a measure of aerobic capacity. And we know that it's related to physiological stress and to dehydration in these birds. Finally, I'm looking at the ratio of heterophil to lymphocytes, and those are two different kinds of cells in birds' blood that you can see by staining a slide and looking at it on a microscope. And the H-to-L ratio provides us information on the immune function of the birds as well as long-term physiological stress. And so by measuring all of these together, I'm hoping to get a good picture of the nestling stress physiology. To provide some proof of concept, blood glucose level is measured kind of on the spot using a handheld glucometer, so we have it to look at. And what you're seeing here is the baseline glucose measure and the stress glucose measure for a set of nestlings. On the y-axis, we have blood glucose level. And what we're seeing is that for almost all of these nestlings, the stress measure is higher than the baseline measure. And this indicates that my nestlings are old enough to have a response to this standardized capture stressor. Interestingly, there's also quite a lot of variation in their responsiveness. And in two cases, I actually see a decrease. And these were, interestingly, two nestlings from the same nest. And it was a nest that had a particularly high load of ectoparasites. So we're seeing some interesting variation in these two nestlings, which are an example of perhaps a dysregulated stress response. The other key measure of my second chapter is parental care behavior. So I am capturing my adult birds here. This is the same one as the nest I showed you earlier, and I'm giving them unique ID bands so that I can tell mom and dad apart. And then I'm taking 3 measures of parental care. I'm recording videos from 8 a.m. to 5 p.m. on 3 different days at 3 different developmental time points. So I've got early development, mid development, and late development, and these are basically different points along the development of endothermy. And then I'm scoring the parent behavior using a software called Boris to get repeat measures of the parent's hourly feeding rate across the day and across different temperatures. So I'm just going to show you an example of one of our nest cams here. So what you saw there was first dad flew in and fed the nestlings, and you can see that he didn't have any colored bands on his legs. And then mom flew in and fed the nestlings, and you saw her bright red bands. And she also took out a fecal sac. So you see that we can tell the parents apart and quantify what behaviors they're performing. My predictions for this chapter for the adaptive responses to urban heat island hypothesis would be that I would see weaker effects of nest temperature on nestling stress level at higher urbanizations than at lower urbanization levels. So as a reminder, I'd expect that high temperatures overall would generally lead to increased nestling stress levels, which corresponds to reduced growth and survival. So if I expect that my urban birds are better able to tolerate higher temperatures, then I would expect nest temperature to have a weaker effect of increasing stress level, and then it would lead to a greater increase at low urbanization levels. I'd likewise expect that nest temperature would lead to less of a decrease in parent feeding rate at my urban nests than it would at my non-urban nests. And that these physiological and behavioral differences in response to temperature would lead to weaker impacts of temperature on reproductive success at my more urbanized nests. So basically, there are physiological or behavioral differences in these urban birds that are helping them cope with the urban heat island. Another part of chapter 2 for me is using a behavioral reaction norm approach to look at individual level variation. So on this slide, I'm talking about population level responses to temperature. I also want to look at how individuals change in their behavior across different temperatures and urbanization levels. So behavioral reaction norm approach looks at this change in behavioral phenotype of an individual over an environmental gradient. And this comes from the idea of reaction norms in quantitative genetics, but here applied to behavior. And it helps us understand the behavioral plasticity within and across individuals across an environment. And we can look at the elevation of this reaction norm, so like the intercept, to get insight into the average value of a trait in an environment. value of a trait in an average environment. And then we look at the slope to understand how plastic or flexible an individual is in its response to the environment. And so behavioral reaction norms are really cool in my opinion because they give us insight into the sources of behavioral variation within and across individuals. So here we have a behavioral phenotype over an environmental gradient again. And I'm showing you a bunch of different potential scenarios. In this first one, we're seeing just one line that increases across the gradient. And so this represents phenotypic flexibility at the population level, but there's no individual variation in the average trait or the degree of flexibility. So there's just one line here. Whereas in the second situation, there's no phenotypic flexibility, but there are consistent individual differences in this average trait. Third, we see, again, there's phenotypic flexibility. Here, there are also consistent individual differences in the average trait, but all individuals show the same level of flexibility. Third, we see differences in the average trait and the level of flexibility individuals show. So now some individuals are more flexible than others. But here you probably wouldn't see overall population flexibility. So like the population would show this flat response, which is why it's super cool to look at the behavioral reaction norms for these individuals. And finally, we see consistent individual differences in the degree of flexibility here, but not the average trait. And these last three cases, where we see consistent individual differences in average trait or the degree of flexibility, these would be caused by either genetic variance or permanent environmental variance. So by genetic variance, I mean variation in alleles that affect the phenotype. And by permanent environment variance, I mean individual-specific environmental variation that permanently affects the phenotype. So this would be like irreversible developmental plasticity. And so it's super cool if we find these patterns because it suggests there are some genetic or developmental differences among individuals that are driving behavioral variation in this trait. And so I want to look at parental care reaction norms across temperature at different urbanization levels. So here's an example of what my results might look like. We're looking at feeding rate as my behavior here on the y-axis and temperature on the x-axis. So here I've got a bunch of different individuals and we're looking at how their feeding rate changes across temperature. And I'd be able to get this because I have so many repeat measures of parental care across times and temperatures for my birds. And what I would predict is that I'd see significant individual differences in the average feeding rate or responsiveness to temperature. So basically my individuals differ in their trait in the average environment, and they also differ in how responsive they are to temperature. And this would suggest some genetic or permanent environmental variation is affecting this behavioral trait. I'd also expect that urban individuals have reduced responsiveness of parental care to temperature. So here I'm suggesting that there's a non-random association of these less responsive phenotypes with the urban environment. And I would expect that this would result in weaker impacts of temperature on reproductive success at my more urbanized nests. Finally, before I finish up, I want to touch on some next steps and significance. By looking at behavioral reaction norms, I can get some very interesting insight into the sources of behavioral variation, but I can't tease apart genetic and developmental or permanent developmental effects. And so I'm really interested in trying to use genomic tools to look at the roles of genetic and epigenetic variation in my birds. So I might look at genomic signatures of selection, for example, using an FST-based analysis, or by looking at the association of my habitat variables or phenotypic traits with genetic variation. I'm also interested in looking at variation in DNA methylation as a mediator of epigenetic effects. So I might look at association of habitat variables or phenotypic traits with DNA methylation level. Or finally, I'm interested in looking at some measures that additionally provide insight into like non-adaptive evolutionary processes like gene flow by looking at population structure and genetic diversity. These are things I'm very interested in, and I have the ability to collect the blood samples to get DNA, but they're pretty out of my wheelhouse. So if you're interested in this and want to talk about collaborating, please reach out. By doing this research, I hope to provide insight into the mechanistic underpinnings of the interactive effects of land use, including urbanization and climate change on organisms that I described before. These two forms of land use change are both having pretty dramatic effects on biodiversity. And to understand how organisms are going to respond to them, we really need studies that investigate how they combine to affect organisms in real world scenarios. I also want to provide insight into the causes and consequences of phenotypic differences between urban and non-urban birds. So we know that in a lot of cases, urban animals show behavioral or physiological or morphological differences from non-urban animals. But we know a lot less about what's causing that variation or what consequences it has for their fitness. So I want to examine sources of behavioral variation along an urbanization gradient to gain insight into the roles of phenotypic flexibility versus irreversible developmental or genetic effects. So I'm using behavioral reaction norms to get at population and individual level variation. And I'm interested in learning about the fitness consequences of differences in behavioral and physiological traits by looking at the reproductive success of my birds and how it's tied to their behavioral and physiological traits in these different environments. With that, I'd like to thank all of the people who've made this project a success so far and who've really contributed to this work, including the Petroselli and Hahn labs, my 2023 field team, including the 2 grad students who collaborated with me on the system, Ian and Jacob, as well as my 5 wonderful undergrad techs. I'd also like to thank the parks departments who've hosted our research, as well as the homeowners who've graciously let us monitor nests on their properties, and the many community members who've contributed in other ways. Finally, I want to thank my funding sources, particularly the SSE, for both the funding and the opportunity to give this presentation. With that, I can take any questions.
Thank you so much, Sage. That was a fantastic talk. I think I could fill in the question discussion with just my questions because our research are so aligned, but I will allow everyone, I guess, to ask questions if they have. So I'm checking the chat. There are no questions there so far, but there are 2 ways that you can ask a question to Sage. Either you just raise your hand and I'm going to try and monitor the screen here, and then you can then just ask your question directly, or you can also paste a question in the chat if you prefer, and then I'll read it, and maybe Robert will help me if I'm missing things, but so far I haven't seen questions on the chat. So yeah, please raise your hand if you have a question. And if I may, I might start with a question of my own.
Sure.
So which one am I going to pick? There is something that you mentioned that surprised me, is that the breeding season of these birds goes from February to August, and that I suppose it means that they have multiple broods. And so in terms of parental effort, since you're interested in this, I was wondering if the strategy of multiple brooding is impacted by urbanization.
Yeah, I'm really curious about that as well. And we are monitoring like both broods for the birds, and I'm interested in looking at whether they're more or less likely to have second broods in urban environments. Just anecdotally, it seems like our urban birds started breeding earlier, and the birds who bred earlier were more likely to have second broods. So I think probably there is something going on there, but I'm really interested to check that out, because it could be that like even if in a single brood, urban birds are fledging less chicks, if they're more likely to have multiple broods, they're actually going to have higher reproductive success.
Yeah, that's what I'm thinking as well. Great. So we have a question on the chat, and I'm not sure if it regards, I mean, which chapter it regards. Harshad might just want to say the question out loud. Else I'm just going to read it. Do you consider the number of nestlings in the nest as a random factor in your analysis? And I'm not, maybe you know which one they mean.
Yeah, I think number of nestlings is probably going to be pretty important for all of my chapters, and it will be included as probably a fixed effect, since the variation is just from like 1 to 5.
Great. Any other question?
Yeah, I just have a quick question. Sorry, I realized I didn't raise my hand.
That's
But
okay.
but great talk, Sage. Thanks for the talk. Really enjoyed it. I just have a pretty simple question. The mortality in the birds that are pre-fledging, how are they dying?
It's a mix of things. Some of them are dying to predators, especially scrub jays. So I'm considering how to deal with that and analyzing the effects of heat at the moment. Some of them we think died as a result of exposure to heat, because after these extreme heat waves, we'd find them either like dried out in the nest or like on the ground under the nest. And we see that when it gets really hot, they kind of try to spread out and they often fall out of the nest onto the ground. So those were the two apparent main causes, were like predators and exposure to high temperatures. We had a couple that we thought ectoparasites, so nest mites, might be playing a role. But yeah, it was mainly those two things, I think.
Got you. Actually, just one more question. If you studied an urban adapted species in particular, like house sparrow or something, do you think you would see the same effect as the great tit study?
I think probably not. And actually, my third chapter is looking at if we take species with different diets or different relationships with urbanization, like if they've been in a city for longer, do we see different effects? So, like, I think that these hypotheses are probably context dependent, and they'll be true. One hypothesis will be true in some species, and one will be
Hmm.
true in another. And so, I'm interested in looking into that context dependence.
So do you mean that you'll have this kind of data across different species?
Yeah, so I'm using the NestWatch dataset, which is like the continent-wide nesting success dataset collected by community members. So I'm using that to look at nesting success across the U.S. in relation to nest temperature and urbanization level.
Mm. So I have a bit of a technical question. I don't know if you have this kind of data. So your analysis, they're based on temperature data that are observed at the nest level. So you call them nest temperature, right?
Mm-hmm.
And it's amazing that you have nest the temperature for all the nests. It's crazy. But I'm wondering if you have an even crazier data set and that you know what the air temperature is. I'm just thinking that in your adaptive hypothesis, part of the adapted behavior of parents might be the way they build or position their nests, in which
Yes.
case you might have differences between air and nest temperatures that are not the same across the urban gradient.
Yeah, actually, that is my fourth chapter that I haven't talked about. I have the logger that's like next to the nest in the air, and then I have the logger that's inside the nest. Or I will in future seasons. I didn't have the logger inside the nest. And then I'm collaborating with some civil engineers to look at the composition of the mud and like the nest itself, as well as the placement of the nest and its dimensions, to see whether the relationship between the air temperature and the temperature the nestlings are experiencing is different across these habitats.
I think that's very cool coming from a person who mainly works on nest box population. You have actually natural nests, and so
Mm-hmm.
you have the natural behavior of building the nest is to look into. There's a new question. Otto, you can unmute yourself if you want to ask it, else I will just read it. So hi, will you measure hyperthermia? And how are you treating hyperthermia as problematic because it develops, considering that tolerance of hyperthermia occurs in songbirds, including facultative hyperthermia?
Yeah, that's a great question. So I think in general, um, it looking at measures of physiology and particularly stress physiology can be complicated because like as a short-term response to an acute stressor, an increase in stress hormones is probably adaptive. I'm not measuring hyperthermia in the nestlings directly, although I am considering trying to measure nestling body temperature across development in future seasons. But when I'm looking at these physiological measures, I'm my theory was that overall, if they're experiencing having to go into this hyperthermic state more often, or if they're experiencing greater physiological or energetic costs of temperature, that's going to reduce their growth, which will reduce their survival. But that's not a given, which is why I'm measuring the growth and survival in addition to the physiological parameters. So I'm going to actually be able to see, is the increase in stress hormone levels leading to the decrease in nestling growth and survival. That's a great question. And like the physiological aspects of this are definitely, I think I'm a behavioral ecologist in training, so it's something that I'm learning about a lot right now.
Great. So I have a bit of an anecdotal naturalist question. You mentioned ectoparasites.
Yeah.
Yeah, so I was wondering if you have differences in prevalence of these parasites between the urban and non-urban areas, and how you
Yeah.
think that plays in.
Yeah, so that one is really exciting to me. So we're counting mainly nest mites on the nestlings, and we count those when we band them. We're also looking at things like blowflies. And so what I saw during the first season was that there were really high mite loads at a lot of our non-urban sites, but there weren't many nest mites at most of our urban sites. And so I think that that might actually be a case where urban birds have an advantage. And I am looking at nest parasite load as sort of one of the features of the biotic environment that might affect these birds' responses to temperature.
Great. Wow, it sounds it. I mean, I already saw that you have a very ambitious project, and then you and then you showed us the slides with your other interests and other things you want to look into. It's very impressive.
Yes, I think I've been told a lot that it'll be hard. I'm going to try to do it. We'll see.
It's good to be ambitious, though. Yes, Harsha, do you want to unmute yourself? Thanks.
Yes, nice talk, Sage. I just had a question. So, when you go to forests versus your urban landscapes, do we always see that there is a high correlation of parasites more in the forested areas? Because that might also lead to your adaptation. And second question is, how often are temperature changes in the forests versus in the city? I mean, if you have a big temperature shift in the city, and if you have a similar increase of temperature in the forest, that might not be equal because you have a lot of other things to buffer it. So there would be, I would expect quite dramatic effects in the buffering capacities of cities or urban landscape versus the forests. So how do we kind of account for it, or have previous studies actually accounted for it? Yes, thanks.
For the first part of your question, I only have like anecdotal reports from my first season, which is that it seemed like parasite loads were higher in our non-urban sites. I think that runs counter to like meta-analyses that have found that parasite abundance and diversity is usually higher in urban organisms. I think it probably depends a lot on your study system, but I am going to look at that and actually model that relationship once I have more years of data. But I wouldn't say that I'm confident in saying it's always that direction at this point. Can you clarify what you mean by cities having, I think you said, greater buffering capacity?
Yes, so in general, I feel that whenever you have a temperature increase, say in a city, I think the effects would be more stark. They would be more directly felt by the birds versus when you have them in a wooded landscape. So even if you have, say, your birds nesting in the tree trunks and all, you would have a lot of foliage to buffer those effects. So there's no screening in the cities as opposed to the forests. So there might be some layers in the forest which actually protect you from heat and those extreme radiation effects as opposed to cities. That's my point.
Yeah, I see. Thanks for the clarification. I think that some of the temperatures we're measuring are right next to the nest. So in some ways they're already being affected by like the canopy cover over the nest, but that's also something that I'm interested in looking at directly, both like very local canopy cover as well as like canopy cover within a couple hundred meters of the nest, because I think that that's pretty important probably in how the parents and nestlings respond to temperature. And sort of I'm regarding it as a measure of their access to these thermally buffered microsites. So I am going to look at specifically the role canopy cover is playing here. If you have ideas for other things to look at, I'm happy to hear them. I've still got 2 more years to go.
No, that's great. That's great. Sure.
Yeah,
Thanks.
you have temperature inside the nest and right outside, and you probably have data on temperature that day in a given area. So that gives you 3 different perspectives. Well, I think it's the hour, so we need to wrap it up. But thanks very much, everyone, for the questions. And thank you so much, Sage, for a really interesting seminar and discussion. And as you may know, everyone, the next GREG seminar is next Monday at the same time.
Thank you, everyone. And feel free to shoot me an email if you have other questions.
Great. Thank you. Have a nice day, everyone. Nice evening.
Great. Thanks, everyone.
Thank
Thank you,
Bye.
Sage.
Thanks for
Thanks, Anne.
hosting.